All right, so we finished off talking about membrane fluidity. Remember that? Membrane fluidity? Yep. All right. So now I want to have a look at some evolutionary adaptations that organisms have to maintain their fluidity. All right, so these are some good real-world examples of organisms that live in perhaps challenging environments and how they have to change the composition of their membranes to make sure the membranes are within the correct fluidity range. Because remember, the function of the membrane is to some degree dependent on its fluidity. All right? Okay, so let's have a look at this sort of case study. So let's think about cold environments, really cold environments. Fish in the Arctic. Okay? So there you've got some fish, and they are actually out in the ice, but they're quite happy out in the ice, and they're quite happy in the water at temperatures that are maybe just a few degrees Celsius. All right? So, freezing cold environments, really, really cold. What's their challenge? What could happen to their membranes in super cold environments? Membranes, cell membranes, they don't change in thickness so much.
It's harder.
Oh, when you said thickness, you were talking about fluidity, right? Okay. All right. So let's try and use fluidity or viscosity. But since I've been using fluidity, we'll talk about fluidity. Right? So what can happen in really cold environments to membranes then? In terms of the fluidity?
Harder and less fluid.
Less fluid, and they can solidify, can't they? A bit like coconut oil in the fridge or cold, it gets solid. All right? So, so all substances undergo density changes, so they can expand or contract when temperature changes. Yeah, and membranes are no exception. Yeah. All right. So their membranes could get solid, not freeze. Right. It's the difference between freezing and going solid. Right. They could solidify at those super cold temperatures. So what's their evolutionary adaptation to prevent that? How could they do it? Sorry?
Is it buffers?
Have a low cholesterol.
So cholesterol works as a buffer. Yeah. So they could modify the cholesterol, and they undoubtedly do that. But what's perhaps the other way they do it? What about the fatty acids? Unsaturated? So what are they going to do to the...
They're going to liquefy it.
It'll be more fluid. Like
But what would they have? What would they...
Now the kinks.
fat. They would be liquid.
I want you to like, you're doing good, but I want you to like describe in a bit more detail rather than just unsaturated fatty acids. You're doing well.
So their chains would be kinked.
They are.
Have that one kind of bond.
But so,
Not
but
entirely fat.
you're right. But your membranes have unsaturated fatty acids in.
They would eat more.
They would have more of the unsaturated
More. They would have a very high proportion of unsaturated to saturated fatty acids. That's what I'm looking for. All right. High proportion of unsaturated phospholipids. All right. You did good, but that's what I want to see, a high proportion. It's the proportion. Okay? All right. Think about another extreme environment then. That's kind of an extreme environment, is it? How does a polar bear or a walrus cope? They have blubber. They're insulation. They don't let their body temperatures drop.
Does that add to...
Sorry?
Never.
Just forget you said anything? Right. All right, can anyone think about another extreme environment then? Right here, Phoenix, Arizona. It's blooming hot, isn't it? All right, let's go to a hot environment then. But we're going to go hotter than Phoenix, Arizona, kinda. All right, so think about bacteria in hot springs. Look at that, gorgeous hot spring in Yellowstone. What are those lovely colours that result from? Yeah, the microorganisms that live there, primarily bacteria. And if we were to have a look at some of those bacteria that live in that hot spring, okay, that's what they look like under an electron microscope. So they have to be tolerant of temperatures at maybe 80 degrees Celsius. Hot, not quite boiling, but super hot. So how do they maintain fluidity within the correct range?
More higher. What was the less?
Begins with P.
Phospholipids. No.
Pro.
Higher pro. Wow.
Pro. Por.
Proportion.
Higher proportion of what to what? Good. A higher proportion of saturated fatty acids. Now, they don't have blubber. All right. Sorry? Oh, you didn't? All right. I thought somebody did. The bacteria don't have blubber, just so you know. All right.
That would make them harder.
Well, blubber will insulate. So if they were colder than the springs, it could help insulate.
So this is just to compensate for their fluidity.
High temperatures are going to make them super fluid, too fluid.
Okay.
So you can compensate by having a very high proportion of saturated phospholipids. But that's not enough.
It's not?
It's not.
Oh man.
They tend to have a whole bunch of other molecules there which sort of help. There are some proteins and cholesterol has a role and so on. But there are other substances, all right, that have a role in making sure the membrane doesn't become too fluid under those high temperatures. Okay? All right. So now what about this environment then? Here's another one. Changing environments. Think about environments, especially for plants, where the temperature changes quite drastically. So winter wheat's a good example. All right. So this is wheat that can grow in the summer, and then it has to be able to endure those high summer temperatures, and then it has to grow through the fall and into the winter, where it can get extremely cold. So its challenge then is in the summer when it's hot and the winter when it's cold. Yeah, those differing temperatures. So what's its solution?
Changing its proportions.
Changing the proportion. Right. So you can change the ratio. In the summer, you might have a lot of saturated, a high proportion of saturated fatty acids. And then as we go into the winter, you're going to slowly replace those with more unsaturated ones in preparation for those freezing winters. Good. All right, you think you get a feel for it? Yep, okay, good. All right, so now I want to have a look at the function of membrane proteins. All of these proteins that are wedged, float around in the membrane, what do they do? What are their different roles? They've got a lot of different roles. All right. Okay, so there are 6 major functions of these membrane proteins. Now I want you to cast your mind back when we looked at a diagram of the membrane structure. And can you describe to me the different membrane proteins in terms of their sort of spatial arrangement or location? Peripheral ones. Yep, what's the other one? Integral ones. Okay, how would you distinguish? And the transmembrane proteins are an example of an integral protein which happens to span the entire membrane. Okay, a little bit peeps out of each side and a whole chunk of it is in the middle. Is everybody okay with that? Yep. All right, so let's have a look. What do they do? Why are they there? Remember, proteins are phenomenally varied molecules, right? Okay, so one is for transport. All right, some of those membrane-bound proteins help move substances into and out of the cells. Okay, that's one role, transport substances in and out. The next one, some of those proteins are enzymes. Enzymes catalyze chemical reactions, as we saw in lab. So some of them are catalyzing reactions. Now, the cell membrane, the plasma membrane, has these enzymes wedged within them. Think about some of the membranes that we've talked about inside the cell, like the mitochondrial membranes, the inner mitochondrial membrane. There are tons of enzymes in that membrane, okay, performing these enzymatic activities. Signal transduction, what that means is that some of these membrane-bound proteins, there's a signal molecule on the outside of the cell which the protein recognizes, and then it's able to sort of the molecule itself doesn't enter the cell, but the signal molecule hits the protein, and then the protein triggers events inside the cell as a result. Is everybody okay with that? It's kind of like I'm standing at the door, right? Somebody comes and whispers something in my ear, then I tell you guys. The whisperer doesn't come in, but I simply pass the message along. Okay? That's what some of them do. All right. Some of them are very important in cell-to-cell recognition. Cells have to be able to recognize other cells. Your white blood cells, for example, go around your body and they're constantly trying to make sure that all the cells that they come in contact with are self cells, not something else, right? It's why you reject organ transplants very often, because your white blood cells are like, oh, red alert, these aren't self cells. And it's why when you have an organ transplant, you've got to match the tissue type. And that way the white blood cells sort of sit and they're like, well, you're mostly similar to self cells, and maybe it confuses them sometimes. And sometimes they send out the red alert message and sometimes they don't. That's why often you have to go on immunosuppressant drugs after an organ transplant. All right, so I'm going to talk more about cell-to-cell recognition in just a moment. Intercellular joining. We've talked about how cells are able to join to other cells. All right? And we've talked about the role of the extracellular matrix. Well, sometimes one cell is attached to another cell as a result of the proteins attaching to each other. The protein in one cell attaches to the proteins in the other, and they kind of lock each other together. Right? And then many of those proteins are attachment points for both the extracellular matrix and the cytoskeleton. So they perform a very sort of mechanical role, not really a chemical one, attachment points. Right. And I'll show you diagrams for each of these in a moment that sort of help you sort of visualize. Good? So they're the major 6 roles. All right, so there we've got transport, and I'll elaborate on these. But you've got these proteins, and they aid in the movement of substances in and out of the cells. Some of the proteins are enzymes. There you've got a substrate, and it gets converted to a product, and that product becomes the substrate of a neighbouring enzyme, which gets converted to a product. And you can have these enzymes next to each other, and substances get converted one into another, into another, into another. Signal transduction. So here's a protein. There's a signaling molecule sent from somewhere else in the body. That protein receives a signal and it sends that message inside the cell so that signal molecule doesn't need to enter. Okay? So we're going to focus on transport a bit later, probably not today, but in the next lecture. All right, cell-to-cell recognition, again, is super important, so I'm going to spend some time and focus on this. So here you've got a protein which has carbohydrates attached to it, which forms the glycoprotein. So very often it's these the shapes of the carbohydrates attached to proteins, which enable the protein in another cell to recognize them or not recognize them. And that's important, and it's a shape fit. All right, so intercellular joining. There's one cell up there and another cell down there, and those proteins are attached to each other, and it helps the cells stick to each other. And again, attachment points to the cytoskeleton on the inside and the extracellular matrix on the other side. Got a good picture then of those membrane proteins, what their roles are? All right, good. So I want to focus on this one. So I'm going to talk about cell-to-cell recognition next, give you a couple of case studies, because they're kind of cool case studies. All right. So first one I'm going to talk about is the HIV case study. An awful lot of research has been done on HIV since its discovery in humans. So you may be aware that HIV sort of made the jump from other primates into humans. It happened in Africa, and we think it happened in the early 1900s. Maybe, and it happened more than once. All right, it made that jump. So it's a virus found in other primates like chimpanzees, and it made the jump. One idea is how it got into humans. Are you aware of what the bushmeat trade is? Bushmeat trade is a horrific practice, really. So lots of parts of Africa, mostly in Central Africa, reliable sources of protein are relatively few and far between, right? Especially in sort of jungly areas. People think of Africa as being these big plains with massive herds of antelope and zebra and so on. So some places are like that, but a lot of places aren't. A lot of places are very jungly, all right? And there's not a lot of large mammals, not a lot of animal protein, all right? And if you go back in time, animal protein was not just rare to see in these areas, but it was hard to catch, all right? So what's happening now is that human populations are exploding, so they're basically expanding out of these little villages, and they're forming more permanent settlements and encroaching out into the jungly areas, all right? And people have got guns. So now there's a very high demand for meat, or at least early in the 1900s, and to some degree still today. So now you've got an efficient way to kill any animal. So a lot of the animals that were out there were monkeys and apes, all right? Now typically they were frightened of people, and there's not many of them anyway, and they're hard to get. But with a gun, you can shoot a lot of them. So what would happen is people would go out into these sort of jungle areas, these outside the village areas. Any animal that moved, they would shoot it. Gorillas, chimpanzees, monkeys, no matter what it was, they'd shoot it. All right? Then they'd go to the animal, they would butcher it, and they would go sell it to people as a source of animal protein. All right? It's called the bushmeat trade. And they would shoot, again, it doesn't matter whether it's a protected or endangered species, they would shoot it and kill it and eat it. All right? But you're in very close proximity to an awful lot of apes. Some of them had the HIV virus in them. You've got a lot of contact with them, a lot of blood-to-blood contact. I think that's how it made the jump one way. It made the jump into humans. All right. So a lot of work's been done about how the HIV virus infects human cells. Okay? And it turns out the HIV virus is a little bit fussy. Not just what species it infects, but what kind of cell it infects. Now remember, a virus is just, in this case, a nucleic acid surrounded by a protein coat. They're not cells, all right? So that virus particle has to be able to sort of know what cell it's come in contact with, all right? So the HIV virus must bind to the white blood cell surface, certain white blood cells in your body, and those white blood cells must have two proteins on them. One is called CD4, and then there's a co-receptor protein called CCR5. As long as those proteins are present on the cell, the HIV virus, the coat is able to recognize them, and as soon as the coat recognizes those two proteins, then it triggers a mechanism and the virus can enter the cell. Okay? If they're not present, the virus doesn't enter the cell, all right? Because it doesn't recognize the cell as being the appropriate cell to infect. Is everybody okay with that?
So are there some people that are genetically immune because they don't have
Yes, there are, and that's what I'm going to talk about. So, if you have both those proteins, you're good. Well, not with respect to HIV. It means that the HIV virus says, oh, you got those, I'm going to infect you. Okay? Now, if you lack either one of those, the HIV virus can't infect you. It's how the HIV virus can recognize human white blood cells, the correct white blood cell. But it just turns out, if you are a resistant individual, you're resistant to HIV because you don't have the CCR5 protein co-receptor. It's just not there. Well, in fact, it is there, but it's not very big and it doesn't stick out. The virus can't find it, if that makes sense. And the reason why you've got this sort of like defective CCR5 protein is because there's a mutation in the gene that codes for that protein. The mutation in the gene codes for a defective protein, which is awesome with respect to HIV. It means you're basically resistant to it. Okay? And it turns out, so a lot of medical research is being done on this CCR5 because some people don't have it, in which case they're resistant. Some people do have it, you're not resistant. So what they think is that the CCR5, it's not a big deal if you've got it or not. Do you know what I mean? It doesn't affect any other part of your biology. Okay? And it just turns out if you're from European descent, then there's a higher probability that you do not have the CCR5 protein. Woohoo for me! Okay, and they think that's linked to the plague that went through Europe. All right, that there was a link there. All right, so if you're resistant to HIV, you're also one of the sort of survivors from the plague, and maybe you survived because you didn't have the CCR5 protein. All right, so that mutation proliferated. All right, so here's the graphic that's in your book. There's the cell, there's the HIV virus, there's the receptor, the CD4 and the CCR5 co-receptor. If they're both there, yup, it can enter. All right. Now, if only one of them's there, it's not access denied, but the virus doesn't recognize the cell as being something it can and should infect. All right. Yep.
So like they say that like if you have HIV that you can have kids that won't have HIV. Is that like where that like
I honestly don't know, because I don't know enough about that, and I didn't know that was the case, to be perfectly honest. I don't know. I can't imagine it's because you don't have the CCR5 virus. I can't imagine that's the case, because you'd have to have a mutation occur in that gene, and I don't think that's the case. If that's the case, the only way I can think it would happen is because somehow you've acquired some immunity from it. I don't know. The baby's had some immunity to it. I don't know.
So if it enters you, but it can't attach itself to any of your cells, what happens to it?
It gets gobbled up by white blood cells or goes away. Doesn't reproduce though. Can only reproduce inside a cell, hijacking the cell's sort of
Systems.
yeah.
Okay.
Yeah.
what all does it do?
So what it basically does is it takes out your immune system. Not completely, but it largely takes it out. So you don't actually die of the virus, but you die of other infections. Your immune system is so shot that other infections that your immune system usually takes care of every day, they take over, and that's what you generally die of. All right, good. It's kind of cool, isn't it? So it shows you the importance of cell-to-cell recognition and kind of how it works. Yep. All right, so I've got an equally cool case. Oh, hang on. So cells then recognize other cells by binding to molecules, and very often the molecules they bind to contain carbohydrates. All right, so in the case of the HIV, it wasn't carbohydrates, it was these proteins. But very often, this recognition process involves carbohydrates bound to the proteins. All right, and it's the specific shape of these carbohydrate chains which is important. So the carbohydrates very often are covalently bonded either to a protein or to a lipid part of the membrane. If they're bound to a lipid, we call them a glycolipid. If they're bound to a protein, we call them a glycoprotein. Now, glycoproteins can have a lot of different roles, but one of them is involved in cell-to-cell recognition. I'll tell you an interesting role of a non-recognition glycoprotein is think about your saliva, how slippery it is, right? It's really slippery. It's because of a glycoprotein you produce in your saliva. It makes your saliva really slippery. All right. So these carbohydrate chains, they can vary tremendously in their shape among species. So you as a species might have different shaped carbohydrates to another species on your cell surface, or even between cells in your body. Some cells have certain shaped carbohydrates, some cells have other shaped carbohydrates. So if you think about it, it's kind of like a labeling process for cells, the shape of these carbohydrates. All right, so here's the next sort of case study I'm going to talk about, and it involves carbohydrates. So have you ever heard of nematode trapping fungi? Yep. Well, that's what we're going to talk about. So these are just too awesome. Here's what happens in the case of nematode trapping fungi. So the fungus set traps for the nematodes in soil. All right, so think about this. In the soil, we've got a whole bunch of microscopic and small organisms. We've got loads of bacteria, loads of fungi, loads of microarthropods, and loads of little worms. All right, all living in the soil. And there are some of these fungi which specialize on trapping and killing nematode worms. All right, but of course, a fungus doesn't have eyes or ears, right? And it's in the dark, in the soil, and it's got to be able to recognize a nematode worm. And it does it as a result of recognizing these carbohydrates on the cell surface. All right? So the fungus must be able to recognize the nematode worm in order for it to catch it, because there's a whole bunch of other stuff in the soil moving around. So membrane proteins called lectins. You don't have to remember the word lectin, all right? You'll probably forget it after this lecture, but membrane proteins called lectins. They recognize carbohydrates on the cell of nematode worms, all right? So think about this then. The nematode worms have got all these carbohydrate chains. That's the identifier for a nematode worm. The fungus has these proteins called lectins, which are able to recognize and bind to those carbohydrate chains. If they recognize the correct shaped chain, that says, gotcha. That says, you are a nematode and I know it. All right? Good? Everybody okay with that? So it takes contact. It takes the nematode worm to literally physically contact the fungus. As soon as it does, and the fungus recognizes those carbohydrate chains, it triggers a whole series of events, and the fungus can trap the nematode. Good? So I'll show you some pictures now how it works. Oh, and then the capture mechanism is triggered. So here's a nematode worm. All right, nematode worms are super abundant worms. Does anybody here have to take 182? You do? All right. If you take it here on this campus, I'm the only one that teaches it, but we do a little section on nematode worms, and they are fascinating parasites. Not all nematodes are parasites, but they have some awesome parasitic life cycles. So pinworms, what have you heard of pinworms? It's a gut parasite you can get. Yep. And so if you ever have kids, right, those who've got kids and they're little and they complain of waking up in the middle of the night with an itchy anus, with an itchy butthole, don't ignore it. All right, because that's what pinworms do. They crawl out at night and they wiggle around, all right, around the anus, and it itches. And of course, what are the kids going to do if they got an itchy anus?
Itch it.
Yeah, scratch it. And that's exactly what the nematode wants, because then the nematodes laid all these eggs around these perianal folds, all right? So now you've got little nematode eggs that are microscopic under the fingernails of the kids. And of course, what do the kids do? Go to school, go to preschool, they're touching every door handle and every surface. And of course, other kids touching the same things, and the eggs go into their gut, and the eggs hatch, more pinworms.
Oh my goodness.
Okay, that's how they spread like wildfire in a school. Okay,
How do you
all
get
right.
rid of them?
Sorry,
How do you get rid
there's
of them?
medications to get rid of them quite easily.
Oh, okay.
All right,
That's weird.
but so there are nematodes everywhere. Not all of them are parasites, but there's a good chance you sitting in the room have nematodes in your body being parasites. All right. So again, in the 182 class, we do an earthworm dissection. And one of the cool things you can do is pluck out the seminal vesicles, which is where the sperm is stored. And it's cool, you get to see worm sperm swimming around, right?
Worm sperm.
The reason why we look in the seminal vesicles is because there's a little parasite that lives in the seminal vesicles that feeds on worm sperm. But sometimes you'll find a little nematode worm also living in the seminal vesicles, feeding on the stuff that's in there. They're everywhere. Some of them are parasites, but some of them, they're just free living in the soil, and they're feeding on just decomposing organic matter in the soil. So that's what these two are. They're nematodes that you would find commonly in the soil, not parasites. Some of them can be parasitic on plant roots. They feed on plant roots, and they're a pest. We don't want them.
So, like, I guess, say you ate dirt when you were younger, played in the mud and got dirty around.
Which you should do, all right?
I
You
don't
should
see
do as a
it. I have
kid.
pictures. Mud buddies,
Good.
mud pies, all that good stuff.
Okay.
So that mean I will likely had with nematodes.
No, if it's a free-living nematode, it's not adapted to be a gut parasite in you.
Oh, okay. So, but there's certain ones that are. That's why you're saying like some are parasites, but there's
Some are parasites and some aren't.
Okay.
Yep.
Makes sense.
Right. So, in fact, if you were out as a kid in the dirt, eating all this stuff and so on, you probably have a rocking immune system. In fact,
Really?
yes,
If you ate dirt as a young kid, you have
absolutely.
My dad, I made a mud pie. I was like, Dad,
I
you
ate
want
a
to try
lot
some of
of
these? Like,
dirt.
I will do. Apparently, I ate it.
I'm not advocating you go eat dirt because there are parasites in dirt you can pick up. If you've got a dog running around your backyard, there's going to be remnants of dog feces all over that backyard. Even if you're not eating dirt, you're just walking around the grass, yep, you're going to pick up dog feces on the bottom of your shoes, and you're going to smother that all over your house and carpet. All right?
So you're pretty much building immunity by being filthy.
But it's not a bad thing, you are. In fact, multiple sclerosis, right? One treatment they're looking at now for multiple sclerosis and some other related diseases is called Helminth therapy. I think it's multiple sclerosis. Anyway,
They eat dirt.
Maybe it's not. But they infect you deliberately with some of these worm parasites. All right? And that's reducing, I think it's multiple sclerosis symptoms.
So I was like OCD when I was a little kid, like, well, still now. But like my grandma would like force me to play in the dirt, and I would just cry the whole time. So like she was doing me a favor.
Absolutely she was, yeah.
I used to run out with them.
I
Yes.
think she was the most awful person in the world.
No. She loves you. That's why she made you play in the dirt. All right. Is anybody obsessed with hand sanitizers?
No.
No? Oh my God. Look to see if you've got any with triclosan in them. Triclosan's an evil, nasty substance. It's going to be banned very soon. It's a lot of hand sanitizers. If it's the alcohol-based ones, well,
My dad's fiance always like sanitize stuff before we eat, and I'm like, I don't know, I feel so alive when I don't.
this whole sanitizing antimicrobial thing has been a push the past 2 decades, and it's because companies want you to buy their products. Right, think about your evolutionary past. You were dirty in your evolutionary past. It meant that you picked up parasites easily, but it also meant that you had sort of different immune systems than what you got today. There are books written about how much of a problem it is that people are so devoid from nature, so worried about getting their hands dirty.
Or like Monk, that show Monk that they used to have.
Yeah.
Yeah.
So when you take those steps back from nature, you end up saying, well, I'm separate from nature. But you're not. You're part of it. And so then when you try and get the bug guy come to eliminate all the bugs, and you literally separate yourself, you're separating yourself from processes and organisms that you rely on for your survival and for the well-being of the planet. Right? It's huge.
Isn't that like where the shortage of bees comes from?
Yes. Overuse of pesticides is another environmental pollutants, is depressing their immune system, so low-level parasites that are there that they could usually take care of, they can't now. It's killing them. All right. So back to the nematodes.
Sorry.
Those of you that are a little obsessed in OCD, there's a whole bunch of things that you can't see and are unaware of that are all over the place. Right? You've got mites right now on your face. You know that, don't you? I've talked about the mites.
Are exploding. And
Yeah.
Stuff.
Yeah. And again, if you have pets at home, oh my God, the level of sort of little creepy crawlies in the carpet, microbes and microarthropods, is off the charts. Just because you can't see them.
Arthropods sound like baby crabs.
All right. So back to the nematodes, living in the soil. Some of these eat plant roots. Oh, we don't like those. All right, agricultural problems. Some of them are just free living. That's where they live. Okay. And here's the fungus. Here's one of the nematode trapping fungi. Fungi grow as filaments, and they make these rings. All right. So these are actually cells of the fungus. This is taken through a microscope, and they make these rings. Now, in order for the fungus to catch the nematode has to swim through that ring. There has to be cell-to-cell recognition, right? The lectins in the fungus have to recognize the carbohydrates in the worm, and there has to be a match, and then it's boom, gotcha. It triggers a capture mechanism, and this happens. The rings. Sorry?
So it's basically just a
Yes. It's like a noose. These swell up, and they catch the worm, and then the fungus grows inside the worm and eats the worm.
So do we like the fungus?
Yeah, we like the fungus.
Heck yeah.
I love fungi. Okay?
You're a fungi.
I am sometimes, yes.
That was really dumb.
Especially after a few days. So here we've got our nematode trapping fungi. All right, these are awesome. In fact, they're being used as a biological control agents for nematodes that are problems on agricultural plants. They eat the roots of the plants. So if you lace the soil with these fungi, then presumably you'll have lower levels of the nematode worms. A much better solution. Maybe it's not quite as effective, but it's a better overall solution than using some of the really nasty chemicals that we add to the soil to kill the worms. Which then, of course, gets into the food that you eat, right? So there's no worms.
Can
But
you
it's
see
not.
the worms? Like,
These nematode worms?
in the plain eye?
Yes, you can. There are some that you need a microscope to see, but these you can see with a plain eye. Yeah. All right. So there's a light this is an electron micrograph. Here's a light micrograph of, again, nematode getting caught because they wriggle, they thrash around, and the fungus doesn't want it to escape. And in fact, the lectins sort of bind quite tightly to the carbohydrates as well.
Was
All
that
right.
airbags?
It does look like airbags, and you can think about it like that. They don't fill with air, though. They fill with liquid. I think they just fill with water. Okay. Some of them have these things called lethal lollipops. So they're adhesive, and when they recognize the nematode, then the nematode sticks to them. But it all comes down to cell-to-cell recognition. All right, everybody good with cell-to-cell recognition then? Yep. Okay, so we're going to move on to this concept number 6, which is diffusion and osmosis. So what I'm going to do now by describing to you what diffusion is I've got to set the stage, because we're going to move on to talk about the movement of substances in and out of cells. Your cells exchange an enormous number of different molecules with the surroundings, with their surroundings. Okay? And everything that moves into a cell, everything that moves out of a cell must pass the cell membrane. There's a lot of traffic that goes in and out of a cell through the cell membrane. And in order to understand it, we've got to understand diffusion. All right? Okay. All right. So here's a good definition of diffusion then. I think we've got to start with this definition. So diffusion is the tendency for molecules to spread out evenly in the available space. All right, so let's think about this for a moment. It's the tendency of molecules to spread out evenly in the available space. All right, so molecules, all molecules are in motion. Now, some of them are moving really quickly. Some of them are just kind of like vibrating a little bit. But I want you to think about gas molecules, for example, and water molecules. They tend to be moving quite quickly, gas molecules especially. All right, so they're just moving around. Are they... Let's just assume there's no wind blowing in the room, right? There's no wind blowing, everything's nice and still. The molecules are moving. Are they moving in any particular direction?
I don't think so.
No. They're moving in random directions. They move around, they move, and they collide and they bounce. They bounce off of things, but they move in random directions. All right? So that's the first thing to think about. Molecules in a gas and a liquid are in constant motion, and they're generally moving in random directions. Unless, of course, I blow, put a fan, or mix the liquid. If it's all nice and still, there's no exterior mixing going on, they're just moving around in random motion in random directions. Okay? Everybody good with that? All right. Now, even though they move in random directions, the diffusion of a population of molecules, like a gas cloud, for example, it might appear to be, and indeed is, directional. So I'll give you this as an example. All right. So if I was to light up a cigar, right? And I'm going to just let the smoke diffuse out of my mouth. Okay? Those molecules, smoke molecules, are in random motion. All right? They're moving around. They're going to try and spread out in the available space. But there is a direction of movement, isn't there? The net movement is away from my mouth, isn't it? And they're going to spread out evenly until they're evenly spread throughout the whole room. Is everyone okay with that? So can you see how random movement can cause movement in one direction? Yeah? At least net movement in one direction. All right. Ooh. Okay. So I'm going to show you a little video that sort of helps you understand diffusion a little bit, but I want to talk about it a little bit more. So I've got my cigar, right? I'm going to puff out the smoke a little bit. I've helped it, yeah? But once that kinetic energy of me puffing out sort of subsided, they're going to do this. In random directions, they're going to spread out evenly in space. They're still moving, okay? And then somebody else walks in the room and they've got a ton of cologne on, let me tell you. And they sit down, yep, and those cologne molecules in random motion are going to move until they're spread out evenly in space. Does the movement, does the diffusion of that cigar smoke have any impact on the diffusion of the cologne molecules?
Will they bounce off each other?
They might. But do they kind of like get to the middle and say, oh, not crossing that line?
No.
No, not at all. The cigar molecules are going to spread out regardless, aren't they? Evenly. Those cologne molecules are going to spread out evenly as well. So the diffusion of one substance doesn't impact the diffusion of another substance. Okay? Is everyone okay with that? They might bounce off each other, they probably do, but they're not going to have any influence about them spreading out evenly in space. Okay, see if we got some audio.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Okay, so there was a whole bunch in that movie that sort of set the theme for what we're going to talk about later, the movement of substances through membranes. Okay, but started off by saying substances spread out in their available space. They move away from the place where they have a very high concentration towards areas where there's a much lower concentration. Is everybody okay with that? So I've introduced this sort of term concentration, right? So let's think about this. Here's that little puff of smoke molecules. Oh my goodness, I've got to tell this, I've got to share this with you as well. It's just, went to Vegas over spring break, went to see Blue Man Group, right? They had drums that they filled with smoke. And they would bang on these drums and these massive circles of smoke would just like come out slowly like this. It was incredible. And then once the kinetic energy had dissipated, the smoke molecules just diffused throughout the room. It was awesome though. So here we've got our cigarette smoke, right, or our cigar smoke. Very highly concentrated. Okay, so let's just say we've got a high concentration, square brackets, over here. And over here, the other side of the room, we've got a very low concentration. In fact, there might not be any of them at all. So the net movement is from an area of high concentration to an area of lower concentration. All right? And these substances are diffusing down their concentration gradient. Okay? They're diffusing down or along their concentration gradient. So is everybody okay with that term, concentration gradient? Yep. And you're okay with this as being the direction of movement. The substance will diffuse from the area where it's more highly concentrated towards the area where it's lower concentration. What happens when the whole room has an even concentration? Well, they just still move, but there's no direct movement. There's no concentration gradient to move along, is there? Because it's all even. All right. All right. So substances then diffuse down or along their concentration gradient. Now, no energy is required or typo there, I don't know what I meant to say. No energy is required for substances to diffuse down their concentration gradients. It doesn't require any energy. It just happens because of the random motion of the molecules. Now you can speed up the rate of diffusion. What do you think is going to speed up the rate of diffusion? I don't know if enzymes would. Heat would. If you heat something, why would heat speed it up? Yeah, when you heat them up, they're moving faster. All right, so it just speeds it up. Okay, but for diffusion to occur, it doesn't require the input of energy. It just happens because the molecules are in constant motion. Okay, so in contrast, you can slow down the rate of diffusion if you cool things down, right? Make them very cold. All right. So some substances then are able to diffuse across the cell membrane. Okay? They're able to cross from one side to the other simply because of diffusion. So molecules are in random motion anyway, but there might be net movement from one side of a membrane, maybe from the outside of a cell to the inside of a cell, if the concentration is higher outside compared to inside, in which case they're going to move through the cell membrane and into the cell simply by diffusion. Some substances are able to do that, not all. Now we'll elaborate on that a lot later, not right now. When we're talking about water. Yep. All right, so let's have a look at this then with respect to a cell membrane. So here's our cell membrane, and a cell membrane is selectively permeable. It lets some substances cross and not others. It lets some substances cross easily, maybe not others. So here we've got a dye molecule, and here we've got water either side of the membrane. And on this side of the membrane, we've added some drips of dye. All right. As a result of the random motion of the dye molecules, and obviously in this case the dye molecules are able to pass the membrane, they will diffuse, net diffusion in one direction until there's an equilibrium reached, in which case there's an equal number going one side as what there is the other. Okay. So at an equilibrium point, what's the concentration of the dye molecules on one side compared to the other? It's the same. Okay. Good. All right. Okay. So let's just say we've got two solutes. Here we've got a cell membrane, which is selectively permeable. We've got water either side. And in this case, this solute can pass through the membrane. So can this solute. Both can. And initially our setup is we've got lots of this solute on the left side and lots of this solute on the right side. Remember, the diffusion of one doesn't impact the diffusion of the other. They can both move by diffusion. This one is going to move to the right. It's going to diffuse down its concentration gradient from the area where it's more highly concentrated to the area where it has a lower concentration, until it's evenly spread out, in which case you've got an equilibrium point. And this substance is going to do the same, except it will diffuse from the right side to the left side until we've got an equilibrium point for both. It's related to the lab that we did. Now in the lab, remember, we only looked at, or mostly looked at, the movement of water. In this case, I'm looking at the movement of a solute. Now it doesn't matter, diffusion applies to water and solutes. Okay? Good? In the lab, we looked at the diffusion of water molecules specifically, which I'll get on to talk about. But generally, I just want you to understand what diffusion is and some of the, I guess, parameters of diffusion. Yep. Because we're going to apply this. Good. So I want you to be really comfortable with diffusion. Okay. All right. So some of these substances that can diffuse across a cell membrane can simply zip through the phospholipid part of the membrane. They can pass right through it, unimpeded. Now, I'll elaborate on that an awful lot more later. All right? But I just want you to be aware that some can move through the cell membrane easily, the phospholipid part, but some, they require proteins to help them move across. All right? And again, I will elaborate on that more later. I just want to sort of illustrate that some things move through the phospholipid part, some things move across the membrane with the help of proteins, but that diffusion alone can drive the movement of substances across the membrane, both into the cell and out of the cell. Okay? All right. Now, if a substance diffuses across a biological membrane, so if a substance moves by diffusion alone, we call that passive transport. And that's because no energy is expended to move those substances across the membrane. It's just diffusion alone that's done it. It didn't require the use of energy to move them. Is everybody okay with that then, that concept? Yep. Substances can diffuse across. Sometimes the phospholipid part, sometimes they need helper proteins. But the driving force that moves them across is diffusion. All right? And they will always move down their concentration gradients. Okay? Good. What makes it passive transport? 2 words. No energy and, I guess 3 words. No energy, doesn't require energy, and diffusion. It's diffusion. Now, diffusion doesn't require energy, but I'd like you to associate those 2 terms with passive transport. It's diffusion, and it doesn't require energy. All right. So let's have a look at this little video clip.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
Okay. Good. So if oxygen moves through a cell membrane, does it need transport proteins, or can it just get through the phospholipid part? It gets through the phospholipid part. And if oxygen was to move from this extracellular space, the fluid, into the cytoplasm, right? What is the process that it moves through? What sort of driving its movement through? Diffusion. All right. And if the net movement is from the outside to the inside, it's because there's a lower concentration inside than outside, and it's going down its concentration gradient. What could be a sink of oxygen? Why could there be a low concentration of oxygen inside a cell? You're using it in what process? Someone said cellular respiration. Good, cellular respiration, yep. So you're continually making a low concentration of oxygen inside the cell because you're using it for cellular respiration. So it's going to continually diffuse across the membrane into the cell. Good? Okay. What about testosterone? Can that get through this phospholipid part? It's a lipid. Totally can slip right through. Lipophilic substances can go right through, like testosterone. Yep. What about glucose? Can that go through? It's large and it's charged. All right?
Large and charged.
Yeah, large and charged. Can't. This repels it. Okay? Sodium ions also can't. They're sort of surrounded by water molecules. It's just repelled. All right. Okay. So now I want to talk about osmosis. Now, osmosis is just a special case of diffusion specifically that refers to water. So osmosis, all the rules of diffusion, all right, apply to osmosis. It's just a special case of diffusion with respect to water movement. So let's define osmosis. Osmosis then is the diffusion of water across a selectively permeable membrane. Water diffuses across a membrane from the region of lower solute concentration to the region of higher solute concentration. That's the net direction of movement until the solute concentration is the same on both sides. And then we're at equilibrium. Yep.
There's a movie called Osmosis Jones. Have you seen that?
You know, I haven't, but I've got to tell you, Is it good? Is it worth watching?
It's a cartoon. Chris Rock plays the main.
All right.
It's about this guy who gets sick and like.
It's Bill Murray. He's in it. So.
Yeah.
Oh, really? All right. Because when I was looking at some, sometimes cruise YouTube and look for some interesting videos to show you, that one came up and I just thought, you know what I thought it was going to be? Sometimes you've got these YouTube videos where someone just yaps and talks. It's like, get to the point, please. And I don't know. I just thought that might be one of them. But it's worth showing?
Yeah.
All right. Okay. You want me to show it, don't you?
It's like an early 2000s like animation movie.
Yeah,
Is
it's
it?
like a kid's movie.
Oh, that's cool.
It's so funny.
Is it good?
It is really fun.
Who's the music?
Yeah.
Who's the music by?
I do not know. It's a movie. So.
Okay. Oh, it's a movie.
Yeah.
Is it very long?
It's not a
Yeah.
music.
It's like an actual movie.
It's like an actual.
Oh,
it's like an
crikey. It's a
hour.
full hour-length movie. Osmosis Jones.
Yeah.
Is it about osmosis?
I don't really
No,
know.
he's
He's blue, but he's like
Oh.
some sort of like cop inside the body of Bill
But it
Murray.
makes
He's
a
like
little
trying to find the virus. that's
Oh,
going through
got it.
victims'
Like,
skin.
Alright.
getting sick, so they're supposed to.
And like there's this cool scene where Bill Murray's got this huge zit and it like pops on Robert Paul's face.
Okay. All right.
Don't they like fight viruses?
Yeah,
like goes in the back.
And then it like hangs out with this pill that comes in and you see it like riding like a roller coaster
Alright.
ride.
I'm into watching that. Maybe I'll watch that.
But with your children.
Yeah,
Alright.
but
Justification.
90s kids in them.
12 and 14-year-old, are they going to say lame? They'll say lame, won't they? 12 and 14-year-old.
I watched it in high school, so.
I
did?
watched it again, but
It's worth it.
Okay.
10
All right.
out of 10 will watch it.
Okay.
Oh.
All right.
An extra credit thing.
So, I want to make sure you understand this.
Yeah, that's interesting.
So, here we've got a solution that has a lower solute concentration than this
Okay.
solution. So, give me an example of a solute.
Something that's dissolved in water.
Sodium chloride is an example.
Is that what it is?
Glucose. Is glucose a solute? Yeah. If it will dissolve in water, it's a solute. Right, the classic ones we talk about, especially with respect to osmosis, are things like sodium chloride. It dissolves and forms sodium and chloride ions. Glucose is another one. It dissolves and it will just form glucose molecules in the water. Good? Okay, so here we've got a solution that has a lower solute concentration than this one, which is a higher solute concentration. Good? So, water molecules then will diffuse from a solution of lower solute concentration across a semi-permeable membrane to a solution of higher solute concentration. Is everybody okay with that? Like, digest it for a minute, mull it over. Okay? When we talk about diffusion, we always talk about the solute concentration. All right. So which direction would water diffuse? If water could move by diffusion from this place to this place across a membrane, which way would it go? From left to right or right to left? Good? Sure?
Yes.
All right, this is what the arrow says. Woohoo! All right. Okay. Everyone okay with that? Because we're going to work on a whole bunch of little problems. But if you can keep this straight, you're good. All right. Okay. So this is a selectively permeable membrane, like a cell membrane. Okay. Over here, we've got side A. Here we've got side B. The solution over here has a lower solute concentration compared to the solution over here. Which direction will water molecules move by diffusion? From A to B. That's what the arrow says. All right. The arrow's always right, yeah? Okay. All right. So that's going to be the net direction. Now, if water molecules move from this side to this side, what's going to happen to the solute concentration here?
It's gonna go up.
It's going to go up, isn't it? Yeah? Because we're losing water molecules. What's going to happen to the solute concentration here? It's going to go down because we're diluting it. Is everybody okay with that? So the net direction of water movement is in this direction. Are there any water molecules moving that way by diffusion?
I wouldn't think so.
Yes. There are. But it just so happens there are many more going in that direction compared to this direction. That's why I say net movement. Maybe 99% of them are moving in that direction and just 1% moving in that direction. Because remember, they're in random motion. All right? They're not saying, oh, everybody go over here. Okay? All right. Good?
So osmosis goes from low to high and from low to
I know what you're about to say. Osmosis, water molecules diffuse from a solution of lower solute concentration to an area of higher solute concentration. Now, I know what you're going to say. You're going to say, well, wait a minute, doesn't diffusion go from high concentration to low concentration? What's the difference?
There's more water. There's more water on that side.
If you, we don't usually think about it in terms of water concentration, right? We don't. But where is the water more concentrated? This side or this side?
Oh, B.
Where is the water more concentrated?
I would say A.
A. So water does follow those rules of diffusion. Water is going from the place where it is more highly concentrated to the place where it is less highly concentrated. Does that make sense?
Yeah.
So that's a common place to confuse, and try not to. We don't usually talk about like the concentration of water. Yep. I could put some numbers on this if you wanted. Let's say look at side A and side B. Let's just say that if we have a high solute concentration here, maybe I've got 10% solute, 10% sodium chloride. In which case, how much water have I got over here? What percent? 90. It doesn't quite work like that, but let's just say it does. Okay, everybody okay with that? So over here, I've got a lower solute concentration. So what's my solute number going to be? Give me a number less than 10.
6.
I'll choose one. I know you guys didn't choose it, but. So what's my percent water? So where is water more highly concentrated? Here. So do you see how it's still going down its concentration gradient? Good? Yep. Okay. All right, I'm seeing some notebooks go away. Not yet. We'll make up all those late times. So we're going to add another 50 minutes on to lecture. All right. All right. I'll tell you what. We'll wrap it up there, though. All right.